All right, neural processing, the retina and beyond. Okay, we're going to get to our primary visual pathways, getting out of the retina. And first I'm going to do this, how did we get here? Which is an allusion to the Talking Heads, just so you know, of that song, How Did I Get Here? So, light reflected from an object into the eye, light focused to form an image of that object on the retina. That light in a pattern that illuminated some receptors intensely, others more dimly, is absorbed by those visual pigment molecules that are packed in the rods and the cones in their outer segments. There are chemical reactions in the outer segments of those photoreceptors, and they transduce the light into electrical signals. As these electrical signals travel through the retina, because we have those photoreceptors, bipolar cells, retinal ganglion cells, and between those two, the horizontal and amacrine cells doing the crosstalk, those electrical signals interact, they excite and inhibit each other. The electrical signals reach the retinal ganglion cells, which because of this processing have center surround receptive fields. Remember, some are excitatory center inhibitory surround, some are inhibitory center excitatory surround. After being processed by the retina, these signals are sent out of the back of the eye in the optic nerve, which this is made up of the nerve fibers, those axons, of the retinal ganglion cells. Okay, so we're here at the optic nerve, and we're going to send that information along some primary visual pathways. First of all, it gets split between the lateral geniculate nucleus and the superior colliculus. I'm going to ignore the kind of what's going to the hypothalamus, but so the majority of signals are going out through the optic nerve, and they are reaching the lateral geniculate nucleus of the thalamus. If you know what the thalamus does, it's really the kind of way station for sensory information. It is organizing, summarizing that information to send it on for higher level processing. And for the vision, that's happening in the lateral geniculate nucleus. Approximately 10% of the signals go to the superior colliculus. So the superior colliculi, we have one in each hemisphere of the brain. They are subcortical structures that are controlling eye movements and other visual behaviors. If you remember me talking early on about the complexity of perception and how difficult it is for us to create a machine that can even navigate its environment. And when we do this with cars, we often use lots and lots of cameras as well as sonar or some other kind of information about where things are, because we cannot get those cameras to kind of move as we have eye movements that are based on what is relevant to me at this time. The superior colliculus also takes in information from other modalities to help control eye movements. As far as, so what goes, most of the information goes to the lateral geniculate nucleus and is sent along then for higher level processing to the primary visual cortex. And I have here the primary visual cortex, the striate cortex. So striate means striped. It has this kind of striped appearance, and they sometimes call it that. Sometimes called V1, meaning primary visual cortex, because just different articles that you're going to read are going to use different nomenclature. From the primary visual cortex, we're going to pass that information along to two pathways, to the temporal lobes for object recognition and to the parietal lobes to help us to navigate in space. One of the aspects of the way visual information is sent to and through the brain is that we have contralaterality. So if you fixate straight ahead, I'm just going to use this example. So if I'm looking straight ahead at a fixation cross in the middle and there's a tree to my left and a house to my right, the left half of each eye is going to receive images from the right visual field. So the left half of each eye is getting that kind of flipped, inverted image of the house. Because remember, I'm looking straight ahead, so it's hitting the left retinas and going back to the left hemisphere. If I'm looking straight ahead and the tree is off to the left, that part of the image, because again it's flipped and upside down, that it is hitting the right half of each retina. And so the right half of each retina is going to send that information back to the right hemisphere. And what you can see is for the kind of outsides of the retina, I'm sending that back ipsilateral, that's to the same side, to the same hemisphere. But for the insides of each retina, I have to send that contralateral. I have to cross hemispheres, and we see that happening at this optic chiasm. So the optic chiasm is right where that X is, where the solid line is passing through the dashed line. And we have, for vision, this kind of complicated contralaterality because it's about the visual field. The lateral geniculate nucleus, again, this is a subcortical structure. It's in the thalamus. Its function is to control the flow of information, to organize information. It's doing some summarizing of this information. I often think of the thalamus and these nuclei as little librarians for us. It's getting input, and I say here at least, from what we understand, it's getting that input from the retina, but it's also getting input from the other nuclei in the thalamus, from the visual cortex, and this kind of top-down processing. The visual cortex is communicating back from itself, from within the lateral geniculate nucleus, and from the brainstem. So when something's really important, I hear a loud noise or something, I would need to move my eyes, or I need to really pay attention and set down those features, that is also input. Whereas its output is primarily to the visual cortex. Okay, primarily to the cortex. So we have this 4:1 ratio of afferents to efferents, where afferents are arriving fibers, or the input, and efferents are exiting fibers, or the output. And it's doing this, it's doing a lot of summarizing of information. Signals arriving to the lateral geniculate nucleus are sorted and organized based on a number of things. One, the eye they came from. Two, the specific kinds of receptors that generated them, so whether it was rods or cones. And then also the type of environmental information they represent. We won't talk a lot about this again, but color information is separate from movement information, all of that in the lateral geniculate nucleus. We've pulled that kind of apart, and we have to put it back together. So one aspect of the lateral geniculate nucleus's organization is that it has this retinotopic map. So A is closer to B than it is to C, both on the cup and saucer and what we see in the retina, right? Because what's up at A is going to hit down at the A prime, or the lower part of the retina. B kind of straight back. C is going to go up. And again, B is closer to C and B is closer to A than A is to C. A and C are far apart. And if we look in the lateral geniculate nucleus, we have maintained this retinotopic map. A is close to B, B is close to C, A and C are far apart. So moving from that picture where A, B, and C are along the cup and saucer, A and C are farther apart, we're going to go ahead and add D here. A and D are even farther apart. A, B, C, and D are points on the retina, basically. And we have this retinotopic map where we have columns that correspond to those particular retinal points, okay? And you can see that we have 6 layers in the lateral geniculate nucleus. We're not going to talk about this very much in this class, but if you take brain and behavior or neuroscience or something, the cerebral cortex, we also see these 6 layers of information, where different information is being kept in those different layers. Okay, and this is kind of a more simplistic map, and I'm going to show another one in a minute. But as we're looking at those columns that are corresponding to particular retinal points, we can see also that we have the blue and the red. The blue, so the rows of 1, 4, and 6, as we kind of shuffle this information in together, that is the contralateral input. So the input from the contralateral eye. And the red, rows 2, 3, and 5, is the ipsilateral input, or the input from the ipsilateral eye, the eye on the same side of the brain. And again, those are amazingly, due to an amazing feat during our neural development, those are shuffled together perfectly so that they match up, and we can have these corresponding points from the retina matching up and meeting each other in the lateral geniculate nucleus. So moving on to the superior colliculus, its function is, overall, what it's doing is it's allowing for eye movement control, and so localization in space is really important. It has these spatiotopic maps, and it is receiving information from different modalities, so different senses. So it also has this kind of multimodal spatial coordination. It's really quite impressive, and again, we really don't understand a lot of how the superior colliculus is allowing us this extra information of our eye movement and letting the brain know where our eyes are looking and putting it all together with other information so that we know where to move our eyes. Okay, so we've gotten to the primary visual cortex, or the striate cortex, our first level of higher level processing when we are in the cortical part of the brain, and not what I keep referring to as these subcortical structures. So I'm going to start with talking about the receptive fields, and I'm going to move on to talking about the organization or the mapping. This is already how we've done things, but one of the things I notice students get confused a lot are what's the difference between these receptive fields, so what the neurons are responding to, and the organization or mapping of these different areas. So we have these different feature detectors. Okay, we have simple cells, which tell us about orientation, complex cells, which tell us about orientation and direction of movement, and end-stopped cells, or hypercomplex cells, that tell us about length, angle, orientation, and direction of movement. We know about these from Hubel and Wiesel, again, doing research on cats, and I'm going to share a video that I think is really fascinating that shows the measuring. So I'm going to show you the measuring of a receptive field of a simple cell in the visual cortex. But first I'm going to talk through the different cortical cells, and then so I can't actually give you this link until after I talk through these different cortical cells. So the simple cortical cell, first of all, if you notice on the left, we have this same kind of, there's excitatory, possibility of excitatory information and inhibitory information. And so I have a vertical bar. Okay, and if you can imagine looking straight forward, and there's a vertical bar hitting, let's say it's in the left visual field, it's hitting somewhere in the right visual cortex. So we have some cell in the right visual cortex that, if you notice at point 0, at this point of the cell, it's hitting vertically. That is where we see the most impulses per second. Once I get off of that vertical 0 point of any kind of oblique, if you kind of imagine how that starts to cross some of that inhibitory space, but if it's not too far oblique off of vertical, it's still hitting a lot of the excitatory space. I, as the simple cortical cell, I'm still going to respond, just not as strongly. And then as we move more and more oblique, where I'm hitting more and more of the inhibitory space and less of the excitatory space, I'm getting towards 0, to where when it gets to horizontal, I'm hitting inhibitory space as much as I am hitting excitatory space, if not more, and I'm at 0. Okay, so I'm basically just coding for, is this vertical, horizontal, or oblique? And if you remember from previous talks, we have more cells coding for vertical and horizontal than we do for oblique lines. If you remember, for the complex cell, it is responding to both orientation of line and direction of movement. So this actually comes with a story where Hubel and Wiesel, they spent some time in the visual cortex and they worked with those dots. You can kind of see on the screen there, those dots, like they were working with in the optic nerve and what they saw in the lateral geniculate nucleus. And they were not getting any response from the neurons in the visual cortex. And they worked at this for a really long time. When you watch that video, I just want to bring up the fact that I usually show about 3 minutes of it in class. I've gone on and watched a little bit more, but I've never probably watched more than 5 or 6 minutes. These guys did this work for hours and hours a day, days of the week, weeks to months to years and years of their lives. And so here they were in the visual cortex working with these dots of light or darkness or whatever they were showing. And one day the cat was anesthetized and hooked up and everything. And they had the neuron hooked up while the slide was coming down. And as the slide came down, it got kind of stuck, and then they heard the neuron go off. I think it was David Hubel, who is quoted in their Nobel Prize winning speech as saying, the neuron went off like a machine gun. It was so exciting to them that this horizontal line that was moving down, that specific orientation in that specific direction was what had this neuron respond. And so they changed their stimuli, they got very excited, and that was the discovery of complex cells. Finally, I'm going to talk about the end-stop cells, sometimes called hypercomplex cells. Just to have you to show what's on this picture, though, you can see the complex cell on the left-hand side, that it is this oblique line. When it's moving up and to the right, when that same oblique line is moving down and to the left, so you see the one with the asterisk, that same oblique line is moving down and to the left. There's no response. It's when it's moving up and to the right. And if we move off of the oblique, so as we move up from that in the picture, we're still hitting some of that, and we're doing something kind of similar. So there's some response in the cell, but really it's the asterisked one where that particular oblique line moving up and to the right, that's what that particular cell is responding to. That's the complex cell. The end-stop cells are, again, just a little bit more complex in that. So if we look at the example on the right, at the very top example, that they're trying to show that this is the part of the figure that is hitting the cell. Okay, so and it's moving this time up and to the left, and if it's just that small portion and it's moving up and to the left, we see some neural firing. If it's moving down and to the right, nothing, right? It's really moving when it's moving up and to the left. And it has to do with orientation as well, but now we're going to see as we move that, so there's more of it, more of that crossing the kind of the receptive field where the asterisk is and the second one down. Now it's a little bit longer, has that same 90-degree angle, moving up and to the left. That's where we see the kind of peak of neural firing, where we see the most impulses per second, like we did in that first figure with the simple cell. Down and to the right, just nothing. We move it farther over that neuron's receptive field, and so it's even longer, and you can see that now we're slowing it down again. So really it's that particular length, that particular angle, moving up and to the left, up and to the left. And I think this is a good place to stop, and I'm going to strongly encourage you to go watch the link that I have, the link within the video, just for 2 or 3 minutes. You might want to watch it for 2 minutes or so and then move in, because it starts with the measuring of a simple cell. That's usually what I show in class, but if you move in a few minutes, they are measuring the receptive field of a complex cell as well. it's really kind of fascinating. And again, remember, these guys spent years of their life doing just this. And just so you know what's happening in that video, the sound is giving you the neural firing.